Welcome back, my dear students. Today we are going to uncover the very important diagnostic technique, radioimmunoassay or RIA. This technique is used for estimating hormones in the body like insulin, thyroxine, cortisol, etc. So we will cover the principal steps, advantages, disadvantages and applications in a clear point-wise way. Today we will look at the principle, steps or procedure, applications, advantages and disadvantages, and after all the clinical significance of the technique. Definition of radioimmunoassay. RIA or radioimmunoassay is an in vitro assay technique that combines antigen antibody specificity with the sensitivity of radioactive detection. Historical notes: this technique was first described for insulin by Rosalind S. Yalow and Solomon A. Burson in the late 1950s or early 1960s. Competitive binding concept: a known amount of radioactive or labeled antigen tracer and the unknown amount of unlabeled antigen from the sample compete for a limited number of antibody binding sites. As the concentration of unlabeled antigen increases, more of the antibody is occupied by the unlabeled antigen. Thus, less labeled antigen is bound to antibody, and thus the measured radioactivity of bound tracer is inversely proportional to the concentration of the unlabeled antigen. After separation of bound versus free tracer, the radioactivity is measured and compared with a standard curve to deduce the unknown concentration. Specific antibody, radiolabeled antigen, sample antigen which is unlabeled, separation method, and gamma or beta counter are the key components of this assay technique. Radioimmunoassay, the diagram shows that it is used to measure concentration of an antigen by use of antibody. The procedure are a known quantity of antigen is made radioactive by leveling it with radioactive isotopes of iodine I-125. Then the radiolabeled antigen is then mixed with a known amount of antibody for that particular antigen. Then antigen antibody bind to each other and the sample of serum from a patient containing an unknown quantity of that same antigen is added. This causes the unlabeled antigen from the serum to compete with the radiolabeled antigen for antibody binding sites. Detailed steps or procedures: stepwise preparation of reagent, antibody of known specificity and concentration, radiolabeled antigen which is hot antigen or tracer, typically using isotopes such as I-125 or gamma emitter or hydrogen 3 or tritium or beta emitter. Unlabeled antigen, that is sample or standard or cold. Buffer, micro titration wells or tubes, wash solution and the separation system. Incubation or competition phase: fixed amount of antibody with the fixed amount of label antigen and sample or unknown antigen or standard known antigen concentration. Incubate to allow competition between labeled and unlabeled antigen for antibody binding. The higher the sample antigen concentration, the more competition, fewer label antigens bound to antibody. Now we should know the separation of bound and free fractions. After incubation, separate the antibody bound antigen labeled or unlabeled from the free or unbound tracer. Methods is the precipitation with second antibody, centrifugation, filtration. Then the bound fraction usually contains the antibody plus antigen complex, and the free fraction is unbound the tracer element. So this is the radioimmunoassay, and the antibody binding sites: antibody, radioactive marked antigen. Radioactive marked antigens and antibody react in a solution. Then unmarked antigens are added to the solution. Then competition between labeled and unlabeled antigens to bind with antibody. Then separation of free unbounded antigens and measure of radioactive antigens bound to antibodies. Measurement of radioactivity: measure radioactivity of either the bound fraction or sometimes the free fraction using a gamma counter or for gamma emitters or appropriate detector. The amount of bound tracer is inversely proportional to the amount of unlabeled sample antigen. The standard curve and quantification. Use standard antigen concentrations known to construct and calibration curve of bound tracer versus antigen concentration. Interpolate the amount samples bound tracer count into the standard curve to determine its antigen concentration. Interpretation: low reactivity bound tracer, high sample antigen, and a high radioactivity bound tracer, that is low sample antigen concentration or negative or very low antigen. Make sure to include controls like zero antigen, maximum tracer binding, and non-specific binding. Applications of RIA. The first of all is the hormone estimation such as insulin, thyroxine, cortisol, testosterone, human chorionic gonadotropin, etc. Drug monitoring, small molecules like drugs, narcotics detection. Tumor markers like peptides, proteins in very low concentration. Viral antigens such as infectious disease, markers early phase in research and diagnostic labs. Research settings where ultra sensitivity or picogram level is required. Advantages: very high sensitivity can detect very low concentration picogram levels, high specificity due to antigen antibody reaction if antibody is good in amount, quantitative and reproducible when done correctly. Useful for analytes present in very low concentration that traditional bioassays cannot easily measure. Disadvantages: involves use of radioactive isotopes, health hazards, requires radiation safety protocols, disposal of radioactive waste, needs specialization equipment like gamma counter, licensed radioisotope handling, and trained personnel. Radio-labeled reagents have limited shelf life like radioisotope decay or instability. The technique has been gradually replaced in many settings by non-radioactive immunoassays like ELISA or ECLIA due to safety and convenience. Potential for non-specific binding cross-reactivity. If antibody are not well characterized, it requires careful optimization. So the summary is that RIA is a highly sensitive quantitative immunoassay using radio-labeled antigen to compare with a sample antigen for antibody binding. Steps are reagent preparation, then competition incubation, further separation, measurement, standard curve, and result. Used widely for hormones, drugs, tumor markers. When very low concentrations need detection. Big advantages is its sensitivity, and big disadvantage is the radioactivity hazard and specialized equipment. So my dear students, today we explored radioimmunoassay helps detect hormones with remarkable precision using the principle of competitive binding between labeled and unlabeled hormones. We saw how its radioactivity is inversely proportional to hormone concentration and how it's applied to measure insulin, thyroxine, cortisol, testosterone, and HCG. Through highly sensitive, it requires caution due to radioactive materials. If you enjoyed learning this powerful diagnostic tool, don't forget to like, share, and subscribe. Stay curious and keep exploring biology. Thank you.